So the manual method of this sequencing, where you need to interpret the gel by yourself, they got the pace with the discovery of automated sequencing, and then within the automatic sequencing, actually the main discovery which helped a lot was the discovery of different dyes which are fluorescent dyes, so that they can give different colors. So what we do here is we take those nucleotides and then we set up different reactions, but primers in each reaction is labeled with different dyes. So these reactions then they don't need to run on different wells on a gel, so we just put them in the same well and they can go on the same lane. And then this band colors, they can help in the detection of which nucleotide is there. So here is how it looks like. So for example, we take the primer for adenine and color it with this green color, and this is T and obviously G and C. So we put them in the same lane, and only the color detection can tell us that which nucleotide is there. You don't even need to compare with the markers. And then these gels are hooked up with some fluorimeters, so that detect the signal or the intensity or the type of the color it's coming out from them, from those bands when they are off from the gels. So they are recorded in special diagrams. We call them electropherograms. Same way in capillary sequencing, there is a little bit modification. So these segments, these DNA fragments, they run through the capillaries. So here is how it looks like. So we have the sample, we put it in a buffer, so that is a negatively charged. Then we have this, we put a capillary in it, right? And then we connect the other end of the capillary to a buffer where there is a positive charge. So DNA runs from this direction to negative to positive direction. So as these bands or these fragments, they go along, there is a fluorescence detector, so it detects which color is there, and then those figures are created in the computers, which we call them as electropherograms. So in this way we can look into these colors, so they can give the idea of what nucleotides are there. So same way the amplitudes of these waves can also tell us about the mutation patterns. We will look into it in the next slides. Here is in comparison, so automated sequencing, where we can take a gel and we can connect to the equipments, and same way we can use a capillary based method, and in the end we come up with something which we call it as electropherograms. These are the charts which represents our sequences. Electropherograms, as I said, they can help us while looking into the pattern of those waves. We can look into the mutations also in the DNA. So for example, we have just one nucleotide we are looking, and some gene is homozygous with respect to that nucleotide. So we can see at position number 3, there is just one big peak coming from this nucleotide T here, since we have T on both sides. In the second situation, which is kind of a heterozygous situation, so we have a T and we have an A. So on electrophoreogram we can have two waves, slightly low amplitude, and then we can say that there is actually a heterozygous situation. If both of them are A's, we can have something like this. So these electropherograms are interesting in terms of detection of those mutations, but obviously now these, in the advanced technologies, there are further advanced techniques to do that. So we conclude that use of fluorescent dyes and an automated system actually improve the efficiency of sequencing.